Good day everyone. Today I will explain what is glycolysis. Glycolysis is a process that breaks down glucose or a simple sugar to release energy. It takes place in the cytoplasm of the cell and can occur with or without oxygen. This process produces ATP, the main energy source for cells. Go. Glycolysis is like the first step in energy production for our cells. When we eat food, especially carbohydrates, they get broken down into glucose. Glycolysis happens in the cytoplasm of the cell and convert glucose into smaller molecules, giving the cell ATP. This ATP is the fuel that cells use to do work like movement, growth, and repair. What interesting is that glycolysis can happen if even if oxygen isn't available. There are the objective. First is to explain what glycolysis is and how it works. This means you will define glycolysis as the process of breaking down glucose or sugar into smaller molecules to release energy. Our first objective is to explain glycolysis itself, what it is, and how it works. Simply put, it is a process that breaks down glucose into smaller parts to release energy that the cell can use. Second, to identify where it happens in the cell. Glycolysis takes place in the cytoplasm of the cell, not inside an organelle like mitochondria. This makes it different from the later stages of respiration. Our second objective is to identify where glycolysis happens, and that is in the cytoplasm of the cell. Unlike other processes that happens in mitochondria, glycolysis happens freely in the cell's fluid part. Third, to describe the main steps and the energy made in glycolysis. Glycolysis happens in the series of steps where glucose is split into two pyruvate molecules. Along the way, it produces two ATP or usable energy and NADH, which helps in later energy steps. Our third objective is to describe the steps of glycolysis. In simple terms, glucose is broken into two pyruvate molecules, and in the process, the cell gains energy, two ATP molecules, and some NADH that can be used later. Fourth, to understand why glycolysis is important for cells and living things. Our last objective is to understand the importance of glycolysis. It is important because it supplies ATP which cells need to function, and it is also the first step of energy production in almost all living things, making it vital for life. In the 80s, scientists studying fermentation noticed how sugar was broken down into alcohol. And in the earlier 90s, researchers began to understand how cells break down sugar to release the energy. And Gustav Emden, Otto Meyerhof, and Jacob Parnas studied and explained the steps of glycolysis. The name comes from the Greek words glykes, or sugar, sweet, and lysis, splitting meaning splitting sugar. And it is also called the Emden-Meyerhof-Parnas pathways to honor the three scientists who studied it.
So where does glycolysis happen and why there? Glycolysis happens in the cytosol, which is the liquid part of the cytoplasm. Glycolysis happens in the cytosol because it doesn't need any special organelles like the mitochondria. And how does glycolysis happen? Glycolysis happens by breaking down one molecule of glucose, or a 6-carbon sugar, into 2 molecules of pyruvate, and a 3-carbon compound. This process makes a small amount of energy in the form of ATP and NADH.
So how does glycolysis happen? Actually, glycolysis has 2 phases, and it is called the energy investment phase, which has 5 steps, and energy payoff phase, which has 5 steps also, equivalent to 10 steps. To further discuss what are the steps, let's have a visual presentation for me to explain further the steps of these 2 phases. So let's watch as these enzymes oxidize one glucose molecule into 2 pyruvate molecules. First is a kinase reaction adds a phosphate onto glucose to form glucose 6-phosphate. This is one of 2 energy consumption steps and is an irreversible reaction. Next is an isomerase reaction converts glucose 6-phosphate into fructose 6-phosphate by rearranging covalent bonds. In step 2 ini, gi-arrange na siya from glucose 6-phosphate, mahimo na dahil siya fructose 6-phosphate by arranging them. Then, another kinase removes a phosphate group from ATP and gives it to fructose 6-phosphate to form fructose 1,6-biphosphate. Kibali, ga-remove ang kinase sa phosphate group nagikan sa ATP tapos ihatag dahil sa fructose 6-phosphate para mahimo na siya na fructose 1,6-biphosphate. This is also the second energy consumption step and an irreversible reaction. The fourth step of glycolysis, allyase reaction splits the 6-carbon fructose 1,6-biphosphate into 2 3-carbon sugars. So, gikan siya sa 6-carbon fructose 1,6-biphosphate na split siya sa ikaduha, which makes it a 3-carbon sugar. Ang pangalan ng 3-carbon sugars or the 2 3-carbon sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The names are actually long. So, the dihydroxyacetone phosphate is rearranged by another isomerase to form a second glyceraldehyde 3-phosphate. So, gi-arrange na sab siya. So, yan na sab from dihydroxyacetone phosphate, nahimo siya sa form na second glyceraldehyde 3-phosphate tungod sa isomerase. At this point in glycolysis, glucose has been metabolized into 2 glyceraldehyde 3-phosphate as I mentioned earlier. So, glucose has been metabolized into 2 glyceraldehyde 3-phosphate and 2 ATPs has been consumed. So the next five steps of glycolysis are energy producing phase. In step number 6, both glyceraldehyde 3-phosphates are oxidized into 1,3-bisphosphoglycerate by a dehydrogenase. This step produces 1 NADH for each oxidized glyceraldehyde 3-phosphate for a total of 2 NADH. These NADH or these two NADH are later used to produce more ATP for the cell to use. Step number 7. A kinase transfers a phosphate from 1,3-bisphosphoglycerate to ADP to form into ATP and 3-phosphoglycerate. This step is also reversible, even though ATP is formed. The next step involves a mutase reaction that moves the phosphate on the third carbon of 3-phosphoglycerate to the second carbon position to form 2-phosphoglycerate. In step number 9, a lyase reaction removes water from 2-phosphoglycerate to form phosphoenolpyruvate. In the final step of glycolysis, a kinase or a kinase reaction removes the phosphate group from phosphoenolpyruvate and donates it to ADP to form into ATP and pyruvate. Like reactions 1 and 3, this step is also irreversible. At this point, 2 pyruvate molecules 4 ATPs and 2 NADH are formed for each glucose that was broken down in glycolysis. The pyruvates and NADH could be used in aerobic respiration to produce more energy for the cell.
So I will discuss where energy or pyruvate go. Net result of glycolysis per glucose molecule has 2 pyruvate molecules, 2 NADH, an electron carrier, and 2 net ATP, 4 made, 2 use, and 2 gain. What happens next depends on oxygen availability. With oxygen, aerobic respiration. Pyruvate moves into the mitochondria, it enters the Krebs cycle, and then the electron transport chain. This produces much more ATP, around 44 or more, giving a total of about 36 to 38 ATP per glucose. So, nagtumusod sa mitochondria, tapos muagi sa Krebs cycle, then mo-produce nga to ng more ATP around 34 or more. Without oxygen, anaerobic respiration or fermentation. Pyruvate is converted into lactic acid in animals, causing muscle soreness after intense exercise. In yeast and some microorganisms, it's converted into ethanol plus CO2. And no extra ATP is produced beyond the 2 ATP from glycolysis. Essential for the next stages of respiration, such as the Krebs cycle and the electron transport chain, where more ATP is made. Without glycolysis, cells would not be able to start the process of extracting energy from glucose, making it a vital pathway for all living things.